Okay, thank you for coming to our research design presentation where we are going to be talking about microglial function and their role in tolerance and opioid-induced hyperalgesia, specifically through the TLR4 receptor. So there are generally two epidemics going on right now in the United States. One is kind of overt, which is the rise in opioid overdose deaths, which has occurred over the past 10 years. And along with that is the epidemic of chronic pain in the US, which opioids are commonly used to treat. And opioids are used to treat pain, and they can have positive effects, which are analgesia or pain relief, but they can also have some detrimental effects, such as tolerance or resistance to the analgesic effects, and opioid-induced hyperalgesia, which we'll refer to as OIH, or an increase in pain because of opioid use. Both of these factors can contribute to dose escalation, which could ultimately contribute to an overdose, which are both negative outcomes. So how do opioids enact their effects? Pharmacological agents, opioid agents, all target the mu opioid receptor to produce analgesia, OIH, and tolerance. But it's kind of unknown how exactly, on a molecular and signaling basis, how opioids enact their effects. Does this occur through glial activation, through specific nociceptor activation? The field is quite divided on how this actually occurs. And in the paper that we chose to focus on, they dissect the role of mu opioid receptors and their location on dorsal root ganglion nociceptors. And they found that knocking out the MOR receptor globally abolished OIH and tolerance, as well as analgesia. And morphine administration led to widespread glial activation in both wild type and MOR knockout mice, which leads them to believe that although morphine activates glia, it does not depend on MOR. So they've kind of resolved that issue in the field that MORs are not found on microglial cells. But it still remains a little bit unclear what the specific role of microglia is. And in particular, we wanted to focus on the last experiment that they performed of conditionally knocking out MOR receptors in the dorsal root ganglion cells of the spinal cord, administering chronic morphine, and a peripheral MOR antagonist, so that all MORs in the periphery were blocked while morphine could enact its effects on the brain. And what they found was that morphine produced analgesia, but none of its negative effects of OIH and tolerance. And our question remains, though, what is the effects of microglia? And specifically, we wanted to investigate the role of the TLR4 receptor, which has been previously implicated in knockout studies and other signaling studies of having effects related to morphine. But the state of the field remains pretty controversial.
So our research questions seek to elucidate the controversial role of microglia in mediating opioid-induced hyperalgesia and tolerance, particularly with respect to the necessity of the TLR4 receptor alongside DRG neurons for nociceptive and anti-nociceptive processes. So our four experimental conditions, which are visualized here, harness both a conditional knockout of mu opioid receptors and a TLR4 antagonist, and we will be observing differences in opioid tolerance and hyperalgesia among these treatment conditions in mice to elucidate the necessity of the TLR4 receptor in addition to mu opioid receptors in mediating analgesia. Based on the results from the literature, we hypothesize that the TLR4 receptor is necessary to maintain analgesia while eliminating OIH and morphine tolerance. So into our research design. So this is our general research paradigm, which breaks down our four experimental conditions. We plan on conditionally knocking out the mu opioid receptor into mice in order to observe the effects of applying an antagonist to inhibit TLR4 receptors, allowing us to observe the analgesic effects of TLR4 in the absence of mu opioid receptors, as well as what happens when both mu opioid receptors and TLR4 are blocked. We will also be looking at the effects of TLR4 antagonists in mice with wild type expressions of mu opioid receptors. We will also be administering a peripheral mu opioid receptor antagonist along with the morphine administration to ensure that all mu opioid receptors in the peripheral nervous system are blocked, and to hopefully recover the same results of previous research, which found that mu opioid receptors regulate analgesia, but not OIH intolerance. Analgesia will be observed through nociceptive behavioral assessments. So first, we harnessed mouse genetic engineering Cree technology and generated conditional knockout mice that lack mu opioid receptors in neurons of the TRPV1 lineage. So as seen here, the knockout mice were generated by crossing mice in which Cree recombinase was driven by the promoter of the TRPV1 gene, with mice bearing alleles in which exons 2 and 3 of the OPRM1 gene were flanked by LOXP sites. So OPRM1 encodes mu opioid receptors, and TRPV1 is a gene specifically expressed in nociceptors in the dorsal root ganglion. So Cree-mediated recombination specifically prevents mu opioid reception in TRPV1 nociceptors for the purposes of our experiment. And then to confirm the knockout of the mu opioid receptors, we will perform PCR on genomic DNA from DRG neurons, the spinal cord, and the brain. And consistent with previous research, we will expect to see a 363 base pair DNA fragment to be excised from the flocked OPRM1 gene.
All right, so after we create the conditional knockout for the mu opioid receptors, we're also going to take two more wild type mice of the same lineage, and we're going to be working to each give its own treatment. So as we already said, two mice will be given an antagonist um, for the MOR receptors, and oh no, sorry. Two will be given an antagonist for the TLR4 receptors, and two will be given a saline injection to leave those TLR4 receptors open so that we can observe their effects when morphine binds to it. So research has shown that naloxone, a drug which actually treats narcotic overdose, can serve as an opioid receptor to TLR4. And basically one MOR knockout and wild type pair will be given the saline solution in order to not tamper with the TLR4 receptors. And then one MOR knockout wild type pair will also be given the antagonist naloxone at a specified dose known to be effective. All right, so after we give the TLR4 either a saline solution or an antagonist, what we are going to do is we are going to be giving a dose of morphine and the peripheral MOR antagonist at a dose kind of that was seen in the Quarter paper to be significant. And we define the MOR peripheral antagonist as methyl naltrexone bromide. And what we will be doing after that is we will be administering that with the morphine subcutaneously, so through the skin in anesthetized mice. The mice will be injected with morphine in this manner once daily for 10 days. And for consistency's sake, we are going to pair a injection of morphine both before and after with the thermal and mechanical nociception assay. So the nociception assays are going to be primarily both mechanical and thermal. And we are going to be able to see the effects of the TLR4 receptors by looking at the differences in responses of the four treatment groups. Following recommendations from the International Association for the Study of Pain in the Quarter et al. Paper, they used a mechanical nociception assay that consisted of applying von Frey filaments of varying forces to the hind paw of the mouse to create a slight bending upon which the withdrawal of the mouse paw was recorded. Also, the thermal nociceptive assay will consist of exposing the mouse tail, 2 centimeters of the tail, to a hot water bath set at a set degree. And after that, the latency to reflexively withdraw the tail will be recorded. Tolerance and OIH will be indicated by a decreased amount of time to withdraw the paw or the tail, indicating either an ineffective dose of morphine or an increased sense of pain. Analgesia will be indicated by increased latency to react.
Okay, potentials and limitations. So as stated before, TLR4 is a protein found on immune cells, and it may have a connected role with MRRs and analgesia and contributes to the negative side effects seen with chronic morphine use. To consider the role of TLR4, we utilize antagonists in combination with knockout and wild-type MRR mice. We expect to see that when TLR4 and MRR are both functional, we will see the negative side effects of morphine use, such as OIH intolerance development. When looking at antagonist TLR4 and knockout MRR, we expect to see an opposite effect where we wouldn't see OIH intolerance development, but we also wouldn't see pain relief. Our expected results will help us to determine how significant TLR4 may be in microglia activation where chronic morphine is involved. Now, when considering future potentials of this technique, TLRs are believed to be implicated in disease, so being able to block the effects of the protein can be beneficial in many ways. If our results do indicate that TLR4 and MRR play a role in OIH and tolerance development, we could block the effects of TLR4 in cases where morphine use is extensively required. A limitation to consider involves the interference or disruption of pathways that may also be connected. In other words, we expect the antagonist to only interact with TLR4, but there may be a chance that it also can interact with an alternative pathway, altering our results. This limitation can also present itself in a clinical setting, making it a possibly unsafe choice for therapy. In the case of our results, we can't even say if TLR4 is the only protein needed to get outcomes such as what was described before. Further experimentation will be needed before anything can be considered conclusive. In combination with our previous steps, our setup, we can then use different doses of morphine to test whether we see OIH intolerance development based on our experimental groups. Using our experimental groups, we expect that chronic administration of morphine will vary between the different groups. With our saline TLR4 and wild-type MRR, we expect to see that with chronic morphine use, mice will be more sensitive to pain, and to overcome that would mean you would need more morphine. But when using an antagonist of TLR4 and wild-type MRR, we expect to see sustained pain relief without the negative side effects present. This will once again emphasize the role of TLR4 that TLR4 has in OIH intolerance development. Now, opioids remain an important drug for those who suffer from severe and chronic pain. If TLR4 antagonist is used in conjunction with morphine use, it will potentially allow for less morphine to be used, and that may lower the dependency for it. Another potential will allow for increased research about the mechanisms of pain and pain tolerance. If we understand more about potential receptors and pathways, that can lead to a better understanding of the effects of opioids and how we can combat such effects. Now, some limitations for our research and technique would mean that rather than seeking out alternative methods for morphine, it may end up increasing the usage of opioids, knowing that we can combat this negative effect. While this knowledge would be helpful, there should still be focus on drugs that are less harmful and addictive all around. Another limitation to be considered would be on the role that morphine has on glial activation. Other studies have indicated a response in glial cells with a focus on microglia. There could be a chance that our technique may be activating other glial cells, resulting in OIH intolerance development, but that's currently not being studied. So more would need to be known before results can be conclusive. So in summary, through the use of conditional knockouts and antagonists in combination with morphine administration in nociceptive assays, we can determine if our hypothesis is supported. Based on our research and results, we can work to develop safer and effective pain management strategies and translate our work into clinical settings by identifying substrates for potential human therapies to treat the opioid-associated symptoms and combat resulting morphine dosage escalation.
So one point, based on kind of taking all this in, that we can kind of go forward with in future experiments, since we already touched that we do not know if other receptors play a role in opioid effects and side effects, we plan that it might be helpful to do a methodological whole genome knockout screening of DRG neurons and microglia and the whole nociceptive pathway using CRISPR-Cas9. And pairing that with phenotypically and/or molecularly identifying signs of OIH tolerance or analgesia in other receptors. And this process, like I said, could be continued down the nociceptive pathway to generate a library of receptors that could be potential targets for treatment of pain.